Hey students, hope you're doing well. In this video, I want to accomplish 3 objectives. The first thing is I want to briefly review membrane structure and function and highlight some of the main things that we've learned so far about the membrane and what it does. The second thing I want to do is I want to describe a process called osmosis, which connects heavily with the membrane and its properties. And we're going to talk about its biological importance as well. And then third, I want to discuss water potential and some practice calculations. This deals heavily with osmosis, and we're going to be doing a lot of calculations in some of our virtual labs dealing with this, so we'll get some practice with that as well. Okay, so first let's talk about the membrane. So what do we know so far about the membrane? Well, we know that the membrane is the outer barrier of a cell. For both prokaryotes and eukaryotes, they have cell membranes. But we also know that in eukaryotic cells, we have organelles that are bound by those membranes as well, right? They're compartmentalized by cell membranes. And so for this first part of Unit 2, after we finish talking about some of the organelles, we're now spending a lot of time talking about the properties of the cell membrane, what is included in it, and really the summary is around this idea of it being a fluid mosaic model. So if you see right here this representation, this term fluid mosaic model really gets at 2 main things about the cell membrane. The first thing is that it is fluid, meaning that all of the components, whether you're talking about the phospholipids or the proteins or cholesterol, that these are not staying in one spot. That if you actually looked under a microscope at a live cell, you would see a lot of these components shifting around in all directions. So you would see phospholipids moving about, although still maintaining that barrier, but there is motion in the cell membranes. It's fluid. The second thing that this model summarizes is that it's a mosaic, right? That there are multiple different components, the primary being these molecules called phospholipids, which we know are referred to as amphipathic, right? Amphipathic meaning that there are hydrophobic and hydrophilic portions of the molecule. That's the primary component. And then within that, you also have these different types of proteins, integral proteins, channel proteins, glycoproteins, all these different things, as well as you have stabilizing molecules like cholesterol, and you have molecules that we'll talk a little bit later in this class, carbohydrates, which oftentimes are involved in being receptors and communicating with other cells, but it is a mosaic, a patchwork of all of these different types of components. Okay, so like I mentioned, the primary component of the cell membrane are these phospholipids, which are represented right here, right? And they're amphipathic, which again means that the tails, which are mainly made of hydrogen-carbon bonds, which are non-polar, they are hydrophobic. They're hydrophobic. And sometimes they have these little bends in one or both tails, and that's actually caused by double bonds. You can check out your group work to see models of why that happens. But the tails, they arrange next to one another because hydrophobic substances like to be with other hydrophobic substances, similar to the hydrophobic R groups in tertiary protein structure. And so we actually see that membranes, they tend to self-assemble into this very organized fashion where we have the hydrophobic tails in the middle and the hydrophilic portions, which again are the heads, and that's mainly because there are some charges, there are some polar bonds due to oxygen and phosphate being present, and so these are hydrophilic. They interact with water, and pretty much any environment you're talking about in terms of a living organism is going to have water, and so these are oriented such that they can interact with water either outside the cell or inside the cell. And when they arrange like this, it literally creates a separation of the outside aqueous solution from the inside, because water can't exist, or is very rarely going to be found, in the middle where it's hydrophobic. Okay, so considering the properties of phospholipids, we then started thinking about how this membrane is known as being selectively permeable. Selectively permeable. That meaning that only certain substances can pass through readily. And a quick way to predict whether or not a substance will be able to move through the phospholipid bilayer is to think about its chemical properties. Typically, if you are a substance that is either polar or charged, if you think ions, for example, that have charges on them, that means you're hydrophilic, right? Water can interact with you. However, in order to make your way from one side to the other, there is a big section that's hydrophobic, and that's going to be very difficult to pass through if you're polar or charged. This is often true for large molecules. It's very difficult for them to move through as well. However, if you are nonpolar and there is no charge, meaning you are hydrophobic, and usually smaller, making your way through this membrane will be a little bit easier because of hydrophobic substances being able to interact with other hydrophobic substances, which is the majority of this barrier. So while substances that are hydrophobic and smaller will be able to move through the membrane easier, there are also proteins that can help facilitate that movement as well, such as channels. And so if a substance is larger, or even if it's polar or charged, we often have channels on the membrane which create literally a channel that's more favorable for that substance to move through the membrane, as opposed to that substance trying to make its way through the membrane on its own. Okay, and just to wrap up this first objective, specifically as we think about how substances move through the membrane, we were introduced to a few different terms. So on the left-hand side of this figure right here, this is passive transport. So passive transport, again, this is transport across the membrane where no energy is required to move the substance. And whether this is simple diffusion or facilitated diffusion, the reason this doesn't take energy is because the substance is going to be moving in a direction that it prefers energetically to move. So in this case, it's always going to be from an area where there's a high concentration to an area where there's a low concentration. The only difference is that when it's facilitated, there's a channel protein or some kind of membrane protein that's allowing a substance to move more readily across the membrane. Okay. Now, the other type of transport is active transport, and this is the type that requires energy, called ATP, which we'll talk about in Unit 3 of how we convert energy into ATP. But this energy essentially allows the process to happen in reverse. So for this particular substance, which is this square substance, this protein is allowing the active transport of the square substance from an area that is low in concentration to an area that is high. And this particular example actually shows the circle substance is being or is participating in active transport as well, just to the other side. So there's a low concentration of the circle substance on this side and a high concentration of the circle substance on the other side. So we have active transport requires energy because it's going in the opposite direction that substances tend to flow. All right, and you also learned just about 2 other examples of active transport that don't necessarily involve proteins like we see in this diagram, but they still require energy. Those 2 categories I'll just put here for your reference were endocytosis and exocytosis. That allows the transport of large amounts of a substance across a membrane, either out of or into the cell. And to just wrap up this first objective, we've talked about how the cell membrane is a selectively permeable fluid mosaic structure in which phospholipids make up the primary components, as well as we see proteins that help facilitate movement across the membrane. All right, so now we're going to move on to our second objective. Okay, so the second objective here is I want to talk about osmosis, describe it, as well as talk about its biological importance. So that's where we're headed right now. So first of all, osmosis, just want to define this, is the diffusion of water across a membrane. And this can be, though not always, facilitated by an aquaporin protein. Because this is diffusion, this is an example of passive transport. No energy is required to do this. So this figure right here summarizes that description. And so what we see here is the plasma membrane. The water molecules are indicated in blue. And on the left-hand side, we see water that is diffusing through the membrane on its own, going from an area where there is high water concentration to an area where there is low water concentration, just like any other substance would diffuse. Now, just a quick note, remember that water is a polar molecule. And I mentioned in the previous portion of this lesson that polar molecules often have a difficult time making their way through the plasma membrane. And that's true for water, although it can slowly diffuse through the membrane. When there's an aquaporin present, which is a protein embedded in the membrane, water is more readily able to diffuse, and osmosis occurs at a more rapid speed because there's a facilitation happening there. Okay, now before we get into more of the details with osmosis, a couple of terms that I want to define here. So the first one is something called a solvent. So a solvent in AP biology is almost always going to be water. This is just the substance in which other substances are dissolved in. And we call those substances that are dissolved in water, we call them solutes. So this is usually substances dissolved in the solvent. Some of the very common examples we're going to see in biology are things like salts or sugar. You can also have other substances like ions, but we'll mainly see salts and sugar. And so if we look at this figure again, right, the solvent is water, and we can think of these red dots as being the solute in this system. Okay, so now that we've described osmosis, we've talked about solvent and solute, I want to talk about more of the process of osmosis. What we're going to see in this class is that osmosis, which is the diffusion of water across a membrane, happens in a specific direction. And what I've added here is that we always got to think about this idea of net movement. Although these figures, right, are showing water going in a specific direction, in reality, we have water molecules going in both directions. It's just that the net, the overall movement over time, is tending towards one side over the other. Okay, so the net movement of water in osmosis, it always occurs in a specific direction. In this class and in biology, this direction is described in 3 ways. I'm going to run through the 3 ways it's described But I want you to keep in mind that all three of these ways are saying the same thing. They're saying the same thing, just in a different perspective for each way. So let's take a look. So the first way, the most obvious way, is that because osmosis is diffusion, osmosis occurs from a high concentration of water to an area where there is a low concentration of water, right? We know that all diffusion is the movement of a substance where there is a lot of that substance to an area where there is less of that substance, right? That's passive transport. Okay, so that way, that's the most immediate way we can think of osmosis, from an area of high concentration of water to an area of low concentration of water. Now, the other way that we can say this, and again, this is saying the same thing, is that we can say osmosis occurs from a region where there is low solute concentration to an area where there is high solute concentration. Remember that solute is a substance dissolved in water, like salts or sugar. And so when we look at this figure right here, right, osmosis is occurring, the movement of water is occurring from a location where there is low levels of solute to an area where there is high levels of solute. All right, remember, just look at the water molecules, and you can see that 1 and 2 here are saying the same thing, just in a different way. Now, we have terms for this. When we compare two sides, in this case of a membrane, the side that has high solute concentration, meaning it has a very high amount of it, we call that a hypertonic environment, meaning that there is a high amount of solute. The other side is called hypotonic, meaning a low solute concentration. So those terms hypo and hypertonic, they refer to the solute concentration. We can say that the net movement of water occurs from an area that is hypotonic towards an area that is hypertonic. Okay, the third way that we can describe the net movement of water is probably the way that's least familiar. And we're going to talk about this in our third part of this lesson. We can say that the net movement of water occurs from a region that has high water potential, which we describe with the symbol psi, to a region where there is low water potential, or low psi. So again, we're going to break down this idea of water potential in just a moment, but just to reiterate, right, all three of these ways of describing how water movement is occurring, they're saying the same thing, just from different perspectives. Okay. Now, to summarize this, let's take a look at a very commonly done experiment to show osmosis. Okay, so this figure is actually from your textbook, and it describes this experiment that's commonly known as the U-tube experiment. And the premise is pretty straightforward. We have this glass tube in the shape of a U, and at the very bottom, in the middle, we have a selectively permeable membrane. This is an artificial membrane that is permeable to water molecules. It allows water molecules to go through, the solvent to go through, but the solute, which in this case is sugar, is not able to pass through that membrane. So this allows us to observe the net movement of osmosis through this apparatus. So what we're going to look at first is take a look at the left-hand side. We have the initial state of this U-tube apparatus. We have water filled up the tube and we have solute, which again are sugar molecules dissolved in the water. Now, if you notice, if you look at the left-hand side of this U-tube, you have a lower amount of solute than you do on the right-hand side. And so right away, we can label this as hypotonic, and this other side is hypertonic, right? Again, that has to do with solute concentration. Okay, so if we were to predict the directionality of water molecules in this setup over time, we know that water is going to move from a region where there is low solute concentration to a region where there is high solute concentration. And so over time, because water can move across this membrane, you're going to see the net movement tend towards the right, and the level of the water on the right-hand side will increase. Now, as that happens, that net movement is going to continue until a form of equilibrium is reached, where there is, in effect, equal concentration of solute on both sides, right? Because if you think about it, you're adding water to the other side, and so you're making that concentration more dilute on the right side. We call that equal concentration, equilibrium, we call that isotonic, right? So osmosis occurs from hypotonic to hypertonic until an isotonic situation is reached. Okay, so to wrap this up, one of the main examples that we can think of when we think about the importance of osmosis is actually how plants get their water, right? We know that plants absorb water through their roots, and then it gets carried up through this structure called a xylem, which is in the middle of their structure. But the majority of that process is actually driven by osmosis, because the net movement that we just described, going from hypo to hypertonic environments, can be thought of in the roots. Roots, which again are made of cells, plant cells, are very often very hypertonic. They're very hypertonic in comparison to the soil. And so when water makes its way into the soil, because of osmosis, water is going to be very quickly absorbed by the cells in the roots because they're hypertonic. There's, in other words, a very big difference in water potential. There's very low water potential in the roots in comparison to the water potential of the soil. And so this helps drive the absorption of water in plants that we're going to talk about a little bit later. So that's one of the major examples that we can think of when we think about the importance of osmosis. All right, so we made it to our last portion of this lesson. What we're going to do is we're going to talk about something I mentioned in the previous section, which was water potential. And we're going to talk about how we calculate water potential, and we're going to run through a couple of practice calculations. Okay, so let's start with just what water potential is. So water potential is it's a type of potential energy that describes the potential energy that water has to diffuse from one area to another. And just keep in mind for this entire section that water will move from an area where it has high water potential to an area where it has low water potential. So what we're going to talk about now is how we actually calculate water potential. This is actually a quantitative value that we associate with water potential, and it's represented with the Greek letter psi. So psi, which looks like a trident, I think of like Poseidon, think of water, Poseidon, can be calculated with two factors. So psi for a water molecule in a particular situation is going to be calculated by adding the solute potential, which is represented by psi subscript s, and the pressure potential, psi subscript p. Okay, so with this equation, we can break this down a little bit more. Most of the time when we look at water potential calculations, we're going to be focused on finding the solute potential. That's usually the information that we're given. And so the solute potential, this portion of the equation, has its own equation. And we calculate that like this. So the solute potential is equal to negative ICRT. Now I'm going to run through what each of these variables mean. Please know that you're not going to be expected to memorize these values and this equation, but I do want you to understand what each component refers to. Okay, so let's start with the I, or the negative I, rather. A lot of students forget that this is a negative I. Please make sure you remember that negative. But the negative I is referring to something called an ionization constant. Okay. Now, what this means is how many ions a solute splits into when it's dissolved in water. Now, there's lots of stuff we could say about this. All you need to know is that when you are dealing with a salt, like sodium chloride, when we dissolve it in water, it becomes sodium ion and a chloride ion. It becomes 2 ions, and so the I value will be equal to 2. That's the ionization constant if the solute is a salt. Now, the other type of solute that we're going to see in this class is a sugar, most commonly glucose, which is C6H12O6. Glucose, for all intents and purposes, is not going to split into different ions. It's going to just remain its one piece when it's dissolved in water, and so its I value will be equal to 1. It doesn't split into 2 separate ions. Okay, so that's what an ionization constant is going to represent. It's either going to be 1 or 2. That just depends on how the solute behaves when it's dissolved in water. Okay, now the C, capital C, is going to represent concentration. All right, and if you've taken a chemistry class, right, you know that concentration is often measured in molarity, or capital M. And molarity is calculated by thinking about the moles of solute divided by the liters of a solvent. Now, we're not going to have to calculate molarity often in this class, but if you haven't taken chemistry, the main takeaway is that concentration is a ratio of solute to solvent, the amount of solute compared to the amount of solvent, which is always water 99.9% of the time in biology. Okay, so that's our concentration. R is going to represent our pressure constant, and this value is always going to be the same. You don't need to memorize this. I'll write it here, but you'll always be given it on any type of exam, especially the AP exam. They give this to you as well. So this value is 0.0831 liter bar moles Kelvin. So there are 2 units on the top and 2 units on the bottom. We're going to see why this is useful for us when we do some practice calculations. But again, it's not necessary for you to know how that is derived. All you need to know is where it goes in the equation. Okay. Last is T, which is temperature. And temperature is going to be in kelvins whenever we're dealing with this equation. So if you haven't taken chemistry, when you are trying to convert temperatures that are given in Celsius to kelvins, the way you do that is you take your degrees in Celsius and you add 273, and that gives you the amount of kelvins for your temperature. Okay. So those are what the variables mean. We have I is the ionization constant, C is the concentration, and R is the pressure constant, and T is the temperature. so we can calculate the solute potential if we have those items. Okay, now that we've talked about the solute potential, just a quick note about pressure potential. Now, oftentimes that pressure potential value will be given to you, and most of the work you'll have to do is around the solute potential. But just for your reference, if you are given a problem where you have an open container, or if you're dealing with an animal cell, more often than not, the pressure potential is going to equal 0 in that kind of system. So if it's an open container, as in an experiment that I'm going to talk about later this week, or if it's involving an animal cell, psi p will be equal to 0. Whenever you have a closed system, a closed container, or a plant cell, this is where you'll often have psi p is greater than 0. And that'll often be given to you. And that is largely due to the plant cell having a cell wall that can withstand pressure building, and a closed container in a similar way being able to hold pressure. So that's just a quick note about pressure potential. Now what I want to do before we wrap this up is just give you an example of a problem that you might see revolving around pressure potential, solute potential, and water potential. Okay, so here's an example of a problem that we might see, and there's going to be two parts to this problem. But first, this problem is going to ask us to calculate the water potential of a 0.2 molar glucose solution in an open container at 22 degrees Celsius. So the first thing is, let's write down our equation here of psi is equal to psi s plus psi p. Solute potential plus pressure potential gives us water potential, and that's what we're solving for, which is psi. Okay, now first thing that we can see from the description is that this is an open container. So right away we know that pressure potential is going to be equal to 0, right? So psi is just going to be equal to psi s, because again, psi p is equal to 0 in this case. All right, so to solve for psi s, we know that this is equal to negative I CRT. Okay, and then we can look at our solution and the conditions to fill in our variables. So first, let's start with negative I. So our I value, because this is glucose, right, our I is going to equal to 1, right, because there's not a splitting up of that molecule when it gets into water. Our concentration, C, is given to us as being 0.2 molar glucose solution. We are also given the constant, which is always going to be the same. It's right here, 0.0831. And then we're going to just copy the units down. It's liter bar divided by mole Kelvin. Okay, and then lastly, our temperature. Right, our temperature is 22 degrees Celsius, but we need to report it in kelvins, and so we just add that to 273, and we get 295 Kelvin. 295 Kelvin. Okay, so we've got our negative ICRT is going to give us our solute potential, and when we calculate that, we get negative 4.9 bars. Now, where do we get those units? So we got to backtrack and look at the units that cancel out. So starting with our concentration, this M, which stands for molarity, is moles over liter. And so when that's going to be multiplied with our pressure constant, the moles are going to be canceled out and the liters are going to be canceled out. That leaves kelvins. So kelvins are going to be canceled out from the temperature since they're on the denominator of the pressure constant. And that leaves bars. So water potential is always going to be given with bars as the unit. In this case, the solute potential, psi s, is equal to the water potential, psi, because the pressure potential was equal to 0. So the answer to part 1 is negative 4.9 bars is the water potential of that solution. Okay, so part 2 is keeping that in mind of the solution inside this open container has a water potential of negative 4.9 bars. If you have an animal cell placed into that solution, predict the net movement of water. So let's first draw a diagram here. Okay, so we know that we have an animal cell placed inside of this glucose solution, which is in an open container. We know that the glucose solution has a water potential equal to negative 4.9 bars. We're also told that the animal cell that we're placing in this solution has a water potential equal to negative 3.2 bars. So we don't need to calculate anything there. But to predict the net movement, we have to remember from part 2 of this lesson that water will move from an area that has a high water potential to an area that has a low water potential. Now this is where keeping in mind the negatives is really important. So in this case, does the animal cell, inside the animal cell, have a higher water potential or lower water potential in comparison to the glucose solution? If you said that the animal cell has a higher water potential than the glucose solution, you got that correct. So that's because we look at those values. Negative 3.2 is a larger value than negative 4.9. So with that in mind, water is going to move out of the cell from an area where there is lower water potential. So water will move out of the animal cell. Okay, so that was just an example of how we can use this to make predictions about the net movement of water. I hope that was helpful, but now at this point we've gone through our 3 objectives, and I hope you have a good rest of your day.